Hi all, you're very welcome to another Saturday session here in ExamRevision.ie. My name's John and I'm going to be looking at Leaving Cert Biology and focusing on the topic of enzymes, with particular emphasis placed on the theory side of enzymes. By the end of today, hopefully you have gained a much greater understanding of enzymes and be able to approach the exam paper questions in enzymes with confidence. So in order to understand the workings of enzymes, we need to understand its role within metabolism. So metabolism is the sum of all chemical reactions that take place within an organism. And essentially, enzymes are responsible for controlling metabolism. They speed up chemical reactions that we're talking about. So for example, at junior cert science, you would have learned about the enzyme amylase, which plays a role in digestion. And amylase is found in the saliva of the mouth. And when we take in starchy foods such as bread or crackers, this amylase acts on the starch to break it down into maltose. If that amylase was not present, digestion would be a lot slower in the mouth. So we're looking at essentially enzymes responsible for metabolism. Why? Because they are catalysts that speed up the rate of chemical reactions, which we'll look at further on in the lesson. When we're talking about the sources of energy in Earth, the primary source of energy for all life on Earth is the sun, so true solar energy. This is very important and a question that actually may appear in photosynthesis. Cellular energy is when we're talking about the breaking down of energy-rich biomolecules in respiration, which releases energy from food. And this energy from the food is used to power all activities within the body. So we're going to get into the nitty-gritty now of enzymes. Just before we do that, what I do suggest you do is get an A4 sheet of paper and make up a small mind map or spider diagram for yourself and label it enzymes. And as we go through this topic, you're going to take down wee points of information that we need to know that I will highlight to you, so that by the end of the lesson, you'll have a lovely condensed set of notes in mind map form that we can directly relate and allow us to help tackle some of the exam paper questions on enzymes. So we're going to start by looking at the enzymes definition. Enzymes are protein catalysts that speed up a reaction without being used up in the reaction itself. So this is really important, and I'm going to put a wee star beside this because we need to know this definition word for word. We don't attempt to put it into our own language. Just want to draw your attention to the third word in that definition. Enzymes are protein, so they're protein in nature. And if we think back to cell structure, and we think of the part of the cell that is responsible for making proteins, we're going to draw the conclusion that the ribosome is responsible for making enzymes, because enzymes are made of protein, and we know that ribosomes produce protein. So just going to draw a wee connection there between your ribosome and your enzymes. So that is the organelle that is responsible for making enzymes, is the ribosome. Proteins are formed by joining the sequence of amino acids together to form a long chain. And if we think again of amino acids, they are building blocks for protein. And they contain the elements, of course, carbon, hydrogen, oxygen, and nitrogen, always. And sometimes they contain phosphorus and sulfur. So again, we're doing a wee bit of revision here as well of our food, because the enzymes and food are directly related. So enzymes are globular proteins, and again, we're going to highlight the fact that they're globular proteins, because our example of a fibrous protein would be keratin, whereas our example of a globular protein are our enzymes. Enzymes have a folded shape, folded into a 3D shape, and what this allows them to do, this allows them to fit the shape of the substrate that they're going to act on. So we're going to look at the analogy of a lock and a key. So once you have this analogy in your head, hopefully it'll make it much more understandable. So I want you to think about the key that opens the car door, the family car door. So that key will open the lock of the door. Perfect. But will that particular car key open the lock for your locker in school? Or will it open the lock that's on your bicycle chain? Or will it open the lock to the local SuperValu? The answer, of course, is that it won't. That particular car key lock will only fit one particular lock. It will not act in any other lock. This is the exact same as enzymes. Enzymes will only act or react with one particular substrate. It will not be able to act on any other substrate. Again, very, very closely associated with how a key works. One key will open one lock, one enzyme will open one or act on one particular substrate. So in the previous slide, we talked about a substrate being the substance that an enzyme will react with. In this situation here, we've got a substrate and an enzyme, and we're going to pretend that this is the key and this is the lock. In this case here, will the key fit the lock? It will fit the lock. So in this case here, the substrate and an enzyme have a complementary or a corresponding shape to each other, which will allow them to fit in neatly and form an enzyme substrate complex, which we're going to talk about further down the line. So this enzyme substrate complex has been formed based on the fact that this shape of the substrate will fit neatly into the enzyme shape here. The part of the enzyme that is responsible for joining with the substrate is called the active site, and later on we're going to associate the active site with being responsible for the enzyme's specificity. The substrate is a substance with which an enzyme reacts, and the product is the substance that the enzyme forms at the end. So again, in visual terms, we can look here, we've the substrate, and if you look at the substrate shape in relation to the active site shape, it fits in neatly again like a lock and our key. And what happens then is an enzyme substrate complex is formed between the active site and the substrate. And again, the whole point or the role of the enzyme is to act on this substrate to produce a product. So when the enzyme and substrate combine, the enzyme acts on the substrate to form products. And the products leave the active site at this point here, and the active site again can now be reused to act on another particular substrate. We're going to look now at some features of enzymes which you need to know for the exam. So enzymes, of course, as we've mentioned already, are made of protein. And at this point, I'm going to use an example of a recall question to ask you what organelle is responsible for producing protein. And hopefully you've come up with the ribosome as being responsible for producing enzymes, because enzymes, of course, are made of protein. I want to draw attention to the way that they may ask you this question in the exam. So frequently the way they may ask a question in the exam, Leaving Cert, is what is the chemical nature of enzymes? Or name the biomolecule from which enzymes are made of. That word biomolecule is going to ring a bell with you because that is essentially what we studied when we looked at food. So this is the type of food from which enzymes are made of, which is your protein. So within the food chapter and biomolecules, you would have looked at our carbohydrates, protein, your lipids, etc. So when they ask you to name the biomolecule from which enzymes are made, essentially what they're asking is what type of food are enzymes made of, which again, we know our answer is protein. Enzymes can function correctly because there's the correct 3D shape to fit the substrate. Again, we're thinking of our lock and our key. One key will open one lock. Enzyme reactions are reversible. This means that enzyme reactions can be anabolic or catabolic. And we're going to focus on this a wee bit further down the line. Enzymes can be named by adding ASE to the name of their substrate. So that's really important. If you see anything ending ASE, generally it's an enzyme. There is exceptions to this, but some of the enzymes we're going to look at down the line now in a few minutes, we will see that they end in ASE. So for example, lipase, again, notice the ASE at the end, that is your enzyme, acts on the substrate lipids. So your lipid there is your substrate. And this is your enzyme, lipase. So when we're concerned in enzymes, we're also concerned with metabolism. And again, we're going to bring a recall question into play here, and we're going to ask, what do we mean by metabolism? the definition of metabolism. So hopefully you have this down your mind map from before and you're repeating this to yourself as I'm asking the question. And of course, metabolism is the sum of all the chemical reactions within the cells of living things or within an organism. So again, in an exam, they may ask you two types of metabolic reactions or two types of metabolic pathways. We mentioned this in the previous slide when we talked about catabolic and anabolic. So catabolic or catabolism refers to the breakdown of larger molecules into smaller molecules releasing energy. Anabolic or anabolism is the building up of larger molecules from smaller molecules using energy. So with catabolic, we want to reference the breakdown. With anabolic, we want to mention the building up. So I want you to use this analogy of a dairy milk bar of chocolate. So a dairy milk bar of chocolate has 8 squares. If I break that bar of chocolate down into 8 separate squares, that is an example of a catabolic reaction. So I'm going one big bar of chocolate into 8 smaller squares. That's catabolic. If I then decide to get 8 of them separate squares and build them back up together into one large bar of chocolate, that's an example of anabolism or an anabolic reaction. So catabolic is we're breaking down, anabolic is we're building up. And again, this is a frequent exam question that may come up. Explain catabolic or explain anabolic. And we need to give these definitions word for word. We need to know examples of anabolic and catabolic reactions in plants and in animals. And again, refer to your mind map where we need to know our definitions of our anabolic and catabolic. So anabolic is the building up of larger molecules from smaller molecules, whereas catabolic is the breaking down of larger molecules into smaller ones. So with that in mind, would we suggest that digestion is building up or breaking down? Of course, we'd associate that with the breaking down. So in animals, digestion is an example of a catabolic reaction because we are breaking down larger molecules into smaller ones. For example, breaking down a sandwich into very small pieces. In plants, our example of a catabolic reaction we're going to use is respiration. And respiration is where glucose is broken down. And the reason why we won't use digestion for plants is because plants don't digest. So there's our example for catabolism in both plants and animals. In anabolism, with animals, we're going to associate protein synthesis as our anabolic reaction. And in plants, we're going to use photosynthesis as our anabolic reaction. Just be very careful with the protein synthesis and the photosynthesis. They're quite alike. So just be really careful of your spelling there with protein synthesis and photosynthesis. Of course, we'll use photosynthesis for plants because animals do not engage in photosynthesis. Okay, that's only our plants are involved in that. We also need to be aware of some catabolic and some anabolic enzymes. So some catabolic enzymes we need to know, and again, we'll link this in later on with digestion. Lipase, amylase, pepsin. There are 3 catabolic enzymes involved in digestion. Lipase acts on lipids. Again, reference the ASE. So lipase reacts on lipids. Amylase acts on starch. And pepsin acts on protein. So we're going to remember the P and P. Pepsin for protein. Later on, we're going to be looking at the effect of pH on enzyme activity. Remember we said enzymes were catalysts that speed up chemical reactions. We do have a couple of things that will affect the ability of enzymes to speed up reactions. And the two we're going to look at is temperature and pH. So I'm just going to reference at this juncture here now that the pH of pepsin is 2. That's the optimum pH of pepsin. So pepsin will work best at a pH of 2. Whereas amylase and lipase, their optimum is between 7 and 8. optimum pH of 7 and 8. And we'll draw back to this information just a wee bit later on in the piece. Anabolic enzymes are examples of DNA ligase and DNA polymerase. And again, we're drawing attention to the ASE at the end of both of them enzymes. So there are anabolic enzymes where larger substances are formed from smaller ones, whereas in catabolic enzymes, their role is to break down larger molecules into smaller molecules. So 2 factors we need to be aware of that affect enzymes' activity. So their ability to carry out their role and their job. What affects them are temperature and pH. You may be asked what is meant by the optimum activity of an enzyme. And the optimum activity of an enzyme is the rate at which an enzyme works best. You may also be asked what is meant by optimum temperature for enzymes. The optimum temperature for enzymes is the temperature at which an enzyme works best at. The optimum pH, again, is the pH at which an enzyme works best at. And this is important, and this comes up very frequently to name 2 factors that affect enzyme activity. So we're going to focus on temperature as a factor that affects enzyme activity. So human enzymes work best at 37 degrees. So we need to maintain our body temperature at 37 degrees at all times, because remember, enzymes control everything. They control metabolism. So we can't have them going out with 37 degrees temperature. And that's why hyperthermia or hypothermia is so dangerous, because our enzymes will begin to lose shape, will become denatured outside of this temperature. Plant enzymes work best at 20 to 30 degrees. And again, you will remember this from your enzyme experiments where catalase was used and the optimum temperature is about 25 degrees. So what will happen enzymes outside of their optimum temperature? And remember what that word optimum means. It means at the rate at which an enzyme will work best. So we want them working at their absolute best. So outside of this temperature of 37 degrees in human beings, enzymes' rate of activity will reduce and they will begin to denature. And denature means that they have lost their shape and they have lost their function. So on our mind map, 2 features of denatured enzymes, they have lost their shape and lost their function. So I'm going to relate this back again to my key and my lock. If I was to melt my key at thousands of degrees, it would change its shape. I want you to think now, will that key work in my lock? If I've melted that key and I let it solidify again back into a solid structure, will the shape have changed? The shape will have changed, of course. Will it now work from my lock? It won't. So what has now happened to that key? It has lost its shape and it now has lost its ability to function. So that's exactly how we describe enzymes' activity outside of their optimum temperature. They begin to lose shape, so therefore their rate of activity will be reduced and they begin to denature. So above a certain temperature, generally we say 50 degrees, enzymes lose their shape completely and become denatured. This is a permanent change in their shape. So at this point now, above 50 degrees, enzymes will not function. And if we again refer to our optimum temperature for human enzymes is 37 degrees, at 50 it makes sense that they will not function properly. Plant enzymes work best at 20 to 30. Over 50 degrees, again, they will become denatured and will not carry out any rate of activity. Where enzymes are involved, graphs are really, really important. You're going to have to draw a couple of graphs potentially in your Leaving Cert based on the effective temperature and the effective pH. So this graph here is looking at temperature and how it relates to plant enzymes and human enzymes. So this is our key up here. So our key tells us that plant enzymes are represented by the green line. Human enzymes are represented by this red line. And I want to just draw attention to the peak of each curve. So I want to draw attention to the peak of the green curve and to the peak of the red curve. And what I mean by the peak is the top, the highest value of each of these curves. So if we're looking at this curve for plants, the green curve, and I want you to pay very close attention to the temperature down here on the x-axis. So we have 10, 20, 30, 40, and 50. And I want you to look at the peak of the green curve and come down to the graph. Come down to the x-axis, and that roughly hits 25 degrees. So the optimum activity of plant enzymes is roughly in around 25 degrees. And you may be asked to explain whether this curve represents plant enzymes or human enzymes. And your answer would be plant enzymes, because the peak of the curve shows optimum enzyme activity at 25 degrees. Whereas if we look at the peak for human enzymes, roughly the peak occurs around 37 degrees. And again, you may be asked to explain whether this curve represents a plant enzyme or a human enzyme. And again, your answer would be a human enzyme, as the optimum level of activity occurs at 37 degrees. From the previous graph, we could see that enzyme activity reaches its optimum in plants and animals at slightly different temperatures. However, we could see that there was a rise in activity as temperature increased. It was more of a sort of a gradual curve. When we're talking about pH, the curve is very, very different. It's a very, very steep, a very sharp upwards and downwards curve. So enzymes work over a very narrow range, and most enzymes work best at a pH between 6 and 8. So again, remember our amylase and our lipase, their optimum pH was in around 7 and 8. So we're going to have another recall question. We're going to think of that enzyme beginning with P that has a completely different optimum pH to lipase and amylase. And this particular enzyme acts on protein, and it's found in the stomach. So hopefully we're recalling that the enzyme I'm talking about is pepsin, which breaks down protein and has a pH of 2, an optimum pH of 2. So outside of their optimum pH, the enzymes begin to lose shape very, very quickly. And remember what happens to enzymes if they lose shape. If they lose their shape, they will lose their function. So we're going to look now at a graph that demonstrates the differences between temperature and pH in relation to enzyme activity. So the first graph we're going to look at is the effect of pH and enzyme activity. Now recall that when we looked at the effect of temperature on enzyme activity, the curves are very much gradual. Whereas with pH, it's up and down very, very sharply. It's a very, very steep roller coaster. Whereas with temperature, the roller coaster is a lot more manageable. It's a lot more gradual. So it's just really important that we understand with pH, there's very little leeway for enzyme activity. It has to be kept within a very narrow range. So for lipase or amylase, our graph is going to be centered on the fact that our peak must be around 7 or 8. We cannot show amylase over here where its peak is a pH of 9 or 10 or 11. Again, you may be asked that what enzyme in the human body is represented by this red curve here. And if you look at this red curve here, its pH is a lot lower. Its peak is at a pH that is a lot lower than 7. So this peak of the red curve, that represents pepsin, where the enzyme activity is at its highest at a pH of in around 2. So just really, really careful when we're doing exam questions and we're asked to draw a graph that reflects an enzyme's activity levels when we're talking about pH, it must be up and down very, very sharply. Whereas with temperatures, it's a much more gradual curve. And this graph just demonstrates the differences between pH and temperature when we're talking about activity levels with enzymes. So with the temperature, we can clearly see that there's a much more gradual curve. There is some activity occurring at 20 degrees here. There is some activity occurring at 30 degrees. The peak, as this diagram shows, for this particular enzyme is in around 40 degrees. At this temperature here, just over 50, how would we describe what's happening to the enzyme? At over 50 degrees, over 60 degrees, we're talking the enzyme is beginning to denature, and over 60 degrees, the enzyme is completely denatured, which means it has lost its shape and it has lost its function. If we compare the pH graph to the temperature graph, we can see that there's a very, very sharp upturn and a very, very sharp downturn. And again, we're referring to the peak as being our optimum pH, where the enzyme, this is the enzyme's pH at which it functions best. So just be really careful with exam paper questions that we're differentiating between our temperature and our pH curve. The pH curve is very, very sharp up and down. The temperature curve is much more gradual. But again, we need to be really careful as to where we show the peak of the curve at. It must relate to its optimum temperature and also to its optimum pH. So during this lesson, we've mentioned the word denatured. So a denatured enzyme has lost its shape and can no longer function. So there's two parts of this. It's lost its shape, and that's number one, and can no longer function. And that's feature number two. And that's the way we may be asked in an exam to name two features of a denatured enzyme. Lost shape and therefore can no longer function. There's two characteristics of a denatured enzyme. So when the shape of an enzyme is fully lost, and this is permanent, it is said to be denatured. It has lost its ability to function. This cannot be reversed. Really important to understand that when an enzyme has completely lost its shape, we cannot change that back. We cannot rehabilitate that enzyme to function again. Denatured is caused by two factors that we're going to look at: unsuitable temperature and unsuitable pH. So we're again relating this back to our key and our lock. If we were to melt that key, it has lost its shape, and that key will no longer open your lock. So it has therefore lost its ability to function. So two important features of denatured enzyme: lost shape and lost function. And this graphic just shows how unsuitable or extreme heat or extreme temperature and unsuitable pH, the effect it can have on an enzyme shape. It has completely denatured the enzyme, it has lost its shape, and therefore it has lost its function. It is no longer going to be able to combine with that specific substrate. So folks, we're going to look now at immobilized enzymes and what they actually are. So immobilized enzymes are enzymes that are attached or fixed to each other or an inert material. What inert means is that basically it won't react with the particular material that it's attached to. So for example, when we did our experiment on immobilization, we would have used sodium alginate gel to attach the enzymes to immobilize them. And again, the enzymes won't react with that particular gel. Bioprocessing is the use of enzyme controlled reactions to produce a product. And bioprocessing will take place in a vessel called a bioreactor. And this is where living things, other products, are used to make a product. So for example, immobilized enzymes are placed in the bioreactor, the substrate is added into the bioreactor, the mobilized enzymes act on the substrate to produce a product, and then the product can be removed from the bioreactor. So again, commercially this is very important, and we're going to look at an application of immobilized enzymes shortly. And in school you would have looked at immobilizing sucrase within your experiment. So just looking at an example of immobilizing enzymes that I mentioned previously, we're going to look at enzymes being immobilized in alginate beads. And that's what we would have used in school. We would have used alginate, sodium alginate gel to trap the sucrase enzyme in wee small beads. We can also again attach or fix enzymes to other materials, particularly inert materials where they won't react with. So some advantages of immobilized enzymes, which comes up very frequently in the exams. Immobilized enzymes can be reused, they can be recovered easily at the end of the process, and they're cheaper. We only need to know a couple of them, so I would pick 2 or 3 and try and learn them off. Again, a lovely question to come up in the exam, that should be very, very feasible to answer. So our application, or our use of immobilized enzymes, glucose is converted into fructose by glucose isomerase. And again, we're looking at the ASE, glucose isomerase, as being the enzyme, and our glucose is our substrate, and fructose is our product. Just another point of information, fructose and glucose are sugars. They end in OSE. So again, I'll put a wee note over here. OSE is our sugar. And remember that ASE is our enzymes. So it's a good way to differentiate between a sugar and an enzyme. So the way enzymes are used in this case is to make a sweeter sugar from a less sweeter sugar. So glucose is relatively sweet compared to other sugars, but fructose is much sweeter. So in order for us to sell more products such as soft drinks, we want that soft drink to be very, very sweet. So what happens is glucose isomerase converts this relatively sweet sugar into a much sweeter sugar, fructose. And fructose, of course, is also in your strawberry jam. Again, another indication of how sweet fructose is. So everything covered up to this point, folks, needs to be understood by both higher level and ordinary level students. We're now going to move on to content that only needs to be covered by higher level students. So I have referred to the active site already within this lesson. And the active site is the part of the enzyme that combines with the substrate. Again, very, very important that we understand this. And this enzyme's active site is responsible for the shape. So if we again refer to the shape of the substrate and the shape of this active site, they're very, very complementary. It'll fit in nicely like your foot into a shoe. So what we want to do is we want to think of our active site on our enzyme as being like a beanbag. And if we were to sit down in a beanbag, it'll sort of mold or alter its shape to fit our shape. And this is exactly what happens with enzymes and their active site. It's not a fixed shape. It's not like the table within your kitchen. If you press down on that table in your kitchen, it's not going to change shape slightly. Because if you press down on a beanbag, it'll alter its shape slightly. And enzymes have the ability to do this because sometimes the enzyme active site is not exactly complementary to the substrate shape. Sometimes the shape can be slightly different. And when they combine, the active site can change its shape slightly to fit this particular substrate. And this is referred to as the Induced Fit Model, also referred to as the active site theory. Either of these will get you the marks in an exam. So before we look a wee bit further into the Induced Fit Model and how enzymes work, we're going to look at enzyme specificity. And again, this is something that I've referenced throughout the lesson. Enzyme specificity means enzyme will only act on one particular substrate. So I would have used the analogy earlier on of the key acting on only one particular lock. So our definition needs to be exact to the point. Enzymes will only act on one particular substrate. And again, if you look at the word within specificity, specific, enzymes are specific in nature because they only act on one substrate alone. The active site is not a fixed shape like that table I'm after referencing. It can alter its shape slightly to fit the shape of the substrate. And again, this is referred to as the Induced Fit Model or the active site theory. And this is very important. The active site is responsible for the specificity of enzymes. Because if we think about what's joining to the substrate, it's the active site of the enzyme that's joining with the substrate. And it has this ability to combine with one particular substrate. And that's where we bring in enzyme specificity. The active site of one particular enzyme will only combine with the substrate, one particular substrate. However, this active site is like a beanbag. It can alter its shape slightly to fit the shape of the substrate. And this is not to be confused with denatured enzymes where their shape changes completely. This is only a temporary shape change when we're talking about the induced fit model or the active site theory. The induced fit model is linked directly with how an enzyme functions or acts, and it's a question that comes up quite frequently. How an enzyme functions, or describe the action of an enzyme. And we'd start off our answer to this question by referencing the fact that an enzyme's active site will only combine with one particular substrate. Referencing our analogy again of our one key only fitting one particular lock. When the substrate joins the active site, we know that it will change its shape slightly to make a better fit. And this is known as our induced fit model or our active site theory. Now, do not get confused with the induced fit model and denaturation. Induced fit model is a temporary alteration to the shape of the active site, and it's only a slight change. Whereas denaturation is a permanent change to the shape of the active site, which will cause it to lose its function. When the active site alters its shape slightly, it allows the substrate to fit with it, and this helps form the enzyme-substrate complex. The substrate molecules react, products are released from the active site, and the enzyme returns to its original shape to be reused. And think back again to what we meant by a catalyst. It speeds up chemical reactions without being used up itself in the reaction. And when we use these enzymes, we can constantly reuse them within our body to help produce products. With a question like this, where we're asked to describe the function or outline the action of enzymes, we put it in bullet point form, where it's much easier for the examiner to correct it, and it's much less complex to read. And again, we need to reference our key features of the action of an enzyme. Enzyme's active site will combine with one particular substrate. Enzyme will alter its shape slightly to fit the substrate. This is known as the induced fit model. These combine to form the enzyme substrate complex. A product is produced and released from the active site, and the enzyme returns to its original shape. If we reference them 5 or 6 points in bullet points, we get full marks in this particular question. This diagram references the lock and key model that I was referring to throughout the lesson, where the enzyme's active site will fit the shape of the particular substrate. However, in some cases, the active site and the substrate may not be exactly complementary to each other. So what happens? The active site of the enzyme will change shape slightly to fit the substrate. In this case, we can see how the active site on image B is different than the active site on image A. So the substrate will fit in nice and neatly to the substrate when it changes its shape slightly. As you can see here, the shape when we form the enzyme substrate complex is different than the initial shape. So we're talking about the induced fit model is a slight alteration to the shape of B into this particular shape where the substrate fits in nice and neatly. So lastly, with reference again to heat denaturation, this is where the shape of the enzyme is permanently changed and therefore it has lost its function. And again, if we recall, 2 features of a denatured enzyme: loss of shape, loss of function. So when enzymes are denatured, we will see no enzyme activity. So if you notice again, over 50, 60 degrees, little to no enzyme activity occurring. Whereas in this case here, we're looking at a peak of roughly just over 40 degrees. This would have been an experiment you would have undertaken in school, where we would have had our denatured enzyme and we would have compared it to control, which is our untreated enzyme, and we would have seen a volume of foam produced within the control, whereas the heat denatured enzyme, we will see no volume of foam produced because the enzyme is denatured. They have lost their shape and therefore they have lost their function. Finishing off the lesson on enzymes, we're going to look at exam paper 2019 question 12 A and B. Between part A and B, this section was worth 36 marks. If you look at section A, there's 9 marks going for this particular part. So what is meant by the term metabolism? And again, this is your definition that we need to give word for word, and then give an example of one anabolic reaction and one catabolic reaction in animals. Now, what is very important here is that you put a star and a highlighter around the word animals, because a key mistake that students will make in this case is they will read one anabolic reaction and straight away they'll put down photosynthesis for anabolic reaction. However, we know animals do not engage in photosynthesis. So therefore, that's a very, very easy 3 marks lost. So our definition for metabolism is the sum of all the chemical reactions in an organism or in the cells of an organism. One anabolic reaction in animals is our protein synthesis, and one catabolic reaction in animals that we would engage in very regularly throughout the day is digestion. And there's 9 marks of the paper that hopefully, after going through this lesson, we would be able to get. Section B is looking at the single most important property of enzymes is their ability to increase the rate of reaction occurring in living things. So this refers to enzymes being catalysts. So our question is asking us, what is an enzyme? And if we think back to one of the first slides in this lesson, we referred to enzymes as protein catalysts that speed up chemical reactions in living things. I want you to really take your time now in this next question. Which type of biomolecule are enzymes? And when we looked at the word biomolecule earlier on, we associated it with food. So essentially what they're asking us there is, what type of food are enzymes made of? And we know that the answer in this case, hopefully, is protein. 2 factors that affect enzymes' action. So if we were to look at enzymes working to their best rate, to their optimum level of activity, what 2 key factors would influence whether they're able to do this or not? So hopefully, again, we're coming to the conclusion that 2 factors are temperature and pH. What is meant by the term specificity in relation to enzyme activity? We're going to underline the word specificity with particular emphasis on the specific part of that word. And we're going to think about our key and our lock. And we're going to refer to specificity, meaning that enzymes will only act on one particular substrate. The next part of the question, part V, is looking for the part of the enzyme responsible for this specificity. So when I'm doing a question like this, I should be underlining all parts of this question, particularly the key parts. So I take my time and I'm looking at the part of the enzyme that is responsible for its specificity. And hopefully you're coming to the conclusion that the part of the enzyme responsible for it being specific is its active site. This is the part where the enzyme will join with. So the key part of this question that I want to talk about is the part VI. And this is the part where you're going to get most of your marks from. Or this is the part where you're going to really achieve particular grade within this question. Because if you look at this question here, it's worth 27 marks. And we break this down and we look at each of these questions. We need to know how much detail to give for part VI. So if we're looking at part I, what is meant by an enzyme, we're going to give 3 marks for that. Which type biomolecular enzymes, 3 marks. Part III is given 2 answers, so that's going to be 3 plus 3, which is 12. And at this point here, we've got 12 marks gone out of the 27. Which means that for these last 3 parts, there's 15 marks somewhere. So what is meant by the term specificity in relation to an enzyme activity? Again, that's very closely related to what is meant by an enzyme. It's a definition. So again, we're going to give 3 marks here. And name the part of an enzyme responsible for its specificity. Again, we're going to give 3 marks. So if I add these up, that's 3, 6, 9, 12, 15 and 18 marks for the first 5 parts of that question, which means that I still have 9 marks to get my full 27. So this last part here is worth 9 marks. And in biology, if anything is worth 9 marks, you must give 3 points. If it's worth 6 marks, we give 2 points. If it's 12 marks, we give 4 points. So generally we will say 3 marks means you must give 1 point about this particular aspect of the topic. So give a detailed account of how enzymes work. So this is us going right back to the start about how enzymes will act on a substrate. So we're going to do this bullet points on our mind map. Enzymes will act on one particular substrate. Enzyme's active site will alter its shape slightly to fit the shape of the substrate. This is known as the Induced Fit Model. Enzyme and substrate will form an enzyme-substrate complex. Products are produced and are released from the active site, and the active site will return to its original shape to be reused. Now in that particular answer for part VI, I'm after giving 5, 6, 7 points, key points of information about this detailed account of how enzymes work. So I have guaranteed to get the marks. So we need to be really, really sure here about how many points we give. There's no need to go overload and give 20 points. Give 5 or 6 nice points to make sure you get them 9 marks. This is the marking scheme of the 2019 paper question 12A and B that we're just after looking at. And just a couple of key pointers I want to refer to. If we look at the definition for metabolism, it's straight to the point. We don't put that in our own words. We learn that exactly as we've learned in our notes or exactly as the marking scheme is looking for. Also, if we look down to specificity, again, our definition can only act on one particular substrate. We do not reuse that word specific in our definition. So we don't say an enzyme acting on one specific substrate. We must use the word particular. Again, if you look at part VI, how do enzymes work? We're putting that in nice bullet points. We're referencing the fact that the active site will change shape to allow for perfect fit. So this is the Induced Fit Model. We're referencing the enzyme substrate complex. We're talking about products being formed and the active site returning to its original shape. So hopefully you did this question. You put the question up on your screen, you paused the PowerPoint, and you were able to get most of these questions correct. Again, we're doing this question in the paper. We underline anabolic reaction in animals, so we do not see protein or photosynthesis here because we're talking about animals. Catabolic reaction in animals, we can use respiration or digestion. So hopefully after that lesson, you have a much clearer understanding of the topic and you'll be much more confident approaching some exam paper questions based on enzymes. Just finishing up, if you'd like to follow us on social media, the following social media handles are on the screen. We're across many different apps and many different websites. And if you have any follow-up questions, please do not be afraid to contact us.